Okay, if we can get started, please. Right, before I go on, any questions on any of the material that we've covered so far? Why are you laughing?
I just loved your comment yesterday about I see a lot of blank.
Well, we've got, if you have problems, don't hesitate to stop by and see me. We also have that review session coming up next week. You know, that's another time we can go over this material. Yesterday we had discussed the organization of the light chains and how a light chain gets expressed. What we want to go to now is the organization of the heavy chain gene family. And again, this is going to be the germline organization of these genes. And then let's see what happens when a cell begins to commit to becoming a B cell, what it does to these genes. All right, so in the heavy chain gene family, and again, remember, all three of these gene families are in separate chromosomes. So this is another group of genes on a different chromosome. Again, you have constant region genes. There is one constant region gene for every class or subclass of heavy chains. So there's 4 subclasses of IgG. There's 4 IgG genes. There's 1 mu. There's 1 delta. There's 2 alphas. There's 1 epsilon and 1 delta. Okay. Now, the heavy chain genes, I don't expect you to remember the order of these things. I do want to point out one thing. And just remember that the first gene is mu and the second gene is delta. Okay. The rest of them, it's irrelevant what the order is, okay? So we have these constant region genes. Now, for simplicity's sake, on this and all the other slides where we're looking at the heavy chains, I'm going to show the heavy chain gene, the constant region gene, as one big exon or one big coding sequence. In fact, each of these constant region genes has a substructure like this. Every domain is actually a separate exon. So, for example, the CH1 domain is one exon. The hinge domain is an exon. Then you have, in the case of mu, you have CH2, CH3, CH4. So every one of these constant regions is actually broken up that each domain is a separate exon. Okay. But again, for simplicity's sake, I'm just going to show it as one box. Okay, because they get to be too complicated if each of these are divided up. All right. So we have these constant region genes. There are variable region genes. And in the case of the heavy chains, the estimates are it's approximately 1,000 heavy chain genes, variable region genes. And each of those variable region genes has a leader region associated with it. You also have J gene segments in the heavy chain gene family. They're sitting there, and there's 4 or 5 of those. But in addition to the J gene segments, there's another small segment called the D segment. And in the case of the heavy chains, it's about 15 of those D gene segments. Again, this is typically we don't refer to it as a gene because it's just a very small, it codes for just a few amino acids. And so we just refer to it as a gene segment. The D stands for diversity. And as we go through today, we'll see why it's called the diversity region. All right. So you have, this is the gene, the organization of these genes in the germline. In a stem cell, any cell that's not a B cell, basically, or committed to be a B cell. So what happens to these genes then when a cell undergoes B cell development in the bone marrow and now commits to becoming a B cell? Well, just like in the heavy chain, in the light chain, it begins to rearrange these genes. But in the case of the heavy chain gene family, there are 2 DNA rearrangements. Okay, here's the germline organization. And just for simplicity's sake, again, I'm just showing just mu and delta. All the other constant region genes are out there. Okay, so what happens is you get DNA rearrangement. And the first rearrangement that occurs is one of these 15 or so D gene segments is brought next to one of the 4 or 5 J gene segments. And again, it's random which ones go. So sometimes you're going to get D1 with J1, sometimes D1 with J2. You get all possible combinations in the B cell populations. Okay. Any one cell, of course, only chooses one D and one J. All right. So you get DJ. So in this example, we're bringing D2 next to J4, and we have now D and J adjacent to each other. And this is the recombination event, and all this material in between that DNA is excised from the chromosome. All right. All right, so that's the first DNA rearrangement. The second DNA rearrangement is then to take one of these 1,000 or so V genes and bring it next to the rearranged DJ. So in this example, we're going to take V2 and bring it next to D2 and bring it next to the D and J. So now we have VDJ all contiguous. And again, it's quotes random as to which V gene is chosen, although there is a bias for those variable region genes closer to the rearranged DJ. But eventually, in the population of B cells, all possible combinations of V's, D's, and J's are going to occur. Again, so so now we have, this is the structure of the DNA, and as a consequence of these rearrangements, again, the gene becomes transcriptionally active for basically the same reason as we saw in the light chain. The enhancer is sitting here in this intron, right upstream of C-mu. Now it's brought close enough, as a consequence of that, the promoters are brought close enough to the enhancer so that this promoter can be active, and you get a transcript. And the primary transcript that you generate from this, it looks like this. It includes the leader, the rearranged VDJ, all of this, and it terminates after C delta. Okay? Now, this primary transcript can then be processed in two different ways. If the primary transcript is processed to remove this intron and then remove this, we will bring VDJ adjacent to C mu. And there's a polyadenylation site where poly A will be put on there. Now you have a message for the mu heavy chain. Alternatively, this message can be processed in this way. Remove this intron and then remove all of this. And there's a poly A site there, and we put poly A. Now we have a messenger RNA for a delta chain. These messages can get translated into protein, and the protein will be transported into the lumen of the endoplasmic reticulum, again, because this leader region has a signal that says, put me in the ER, so it goes to the ER. And now you have, and in the process, of course, the leader is clipped off, and you end up in the ER with a heavy chain for either with a mu chain or a delta chain. Those can reassociate with whatever light chain this B cell is making, and then display on the membrane a mu and a delta. All right? Now, notice, even though we're expressing two heavy chains, a mu and a delta, the specificity is the same. The reason is it's the same VDJ region being used here as is being used here, and that combines with whatever light chain that cell is making, so the specificity of the two antibodies are the same. So mature B cells end up with, on their surface with an IgM and an IgD of the same specificity. Right?
Talk about J4, J5.
J4?
Out there you've got J4 and J5.
Right.
And then in the next picture, right below that, you just got K.
Oh, obviously I just tried to, it was getting too close together to put all that. That is J4, right? You mean in here, going from here to here? Mm-hmm. Yes, what's happened is, of course, J5 is deleted out because of the recombination event. The recombination occurs from here to here, so J5 is removed. All right? So this is actually D2, this is J4, and that's C delta. Okay? And it's also here, right. I mean, what's happened here is this is removed, this part here. So the J5 is being removed. So this becomes J4. I just didn't have enough room to stick all the subtitles, the subscripts in there. Okay? Any other questions?
You mentioned about IgM and IgD, that just have one constant. How can we have IgM and IgD?
I say IgM and IgD. D, right? Because this is the delta region, constant region, okay? That's coded for by this gene. This becomes this. This is the mu chain, and that becomes that. So you have different constant regions. But the variable regions, the VDJ, is the same. That is not changing. That's the same one up here. It's just being put on different constant regions, okay? So the point I was making is that when this heavy chain associates with the light chain, whatever variable region is present on the light chain and whatever variable region is present on the heavy chain will determine the specificity, because those are the regions that contain those hypervariable regions that create the pocket for the antigen. So the specificity, the antibody combining capacity of these is the same, although they're two different classes. And so that's why B cells have on their surface these two classes, because of this differential RNA processing, the way you can process this primary transcript. Okay? Any other questions? Yeah.
So here you've got IgG and IgM. Those would just be kicked out at random over time, but in a population you have the same specificity. In other words, they don't come out at the same time.
You mean the mu and the delta? In fact, the mu comes a little before the delta. There's some preference to process the message to make this kind of change. So early on, you do see mu, but eventually it also starts processing this way and makes delta. So you do get both of them made eventually. And that's why B cells, mature B cells, have on their surface both of those classes of immunoglobulins, M and D. Okay? Any other questions? All right. So how do these rearrangements occur? This is a recombination event. And while it uses some of the normal recombination machinery within the cell, there are some very unique features of this recombinase that carries out this recombination. What this recombination is dependent upon is the recognition of things that have been called recombination signal sequences, or RSSs. And these have a unique structure. They all contain this heptamer, a sequence of 7 nucleotides, and an ionomer, a sequence of 9 nucleotides. And those are conserved sequences, but they are separated. The heptamer and the nonomer are separated either by 12 bases or by 23 base pairs. And it doesn't make any difference what the sequence of those 12 or 23 is. What's important that it be 12 nucleotides or 23 nucleotides. Now, the reason that's important is that when you take 12 nucleotides, what will happen? 12 nucleotides is what it takes to move one time around the helix, okay, of the double helix of the DNA. So consequently, that results in the nonomer and the heptamer being on the same side of the DNA molecule, all right, when it's in its helical form. 23 base pairs is what it would take to go around the helix twice and bring the nonomer and the heptamer on the same side of the sequence. So basically what's happening for this recombination event, these signal sequences, the heptamer and the nonomer, have to be on the same side of the DNA molecule. Those signals that involve, they have 12 base pairs are simply just referred to as one-turn RSSs, and the 23 base pairs are referred to as two-turn signal sequences. Now, in this recombination event, there's only recombination between a one-turn signal and a two-turn signal. So the recombination only occurs between a one-turn signal and a two-turn signal. Now, if you look at the light chains, and if you look at the way the genes are organized and where these signals are located in the intron immediately after the V gene, in the case of the lambda light chains, and immediately upstream of the J gene segment. And when you look at them and you analyze them, what you see is that after the V gene, you have a two-turn signal, and right before the J gene, you have a one-turn signal. That ensures that there's always going to be a V brought next to a J. You could never bring V next to C because there's no signal. There's no recombination signal right in front of the C gene, okay? So it always ensures a V to J joining, because you have to obey this one-turn, two-turn signal. You have to employ both the one-turn and the two-turn signal in the recombination process. In the kappa genes, the genes are organized just the opposite. After the V gene, you have a one-turn signal. In front of the J gene, you have a two-turn signal. But again, you can only go V to J. You can never skip the J. You always have to pick a J gene segment in this recombination, because you have to recombine a 1 and a 2. And the heavy chains are organized like this. After V, there's two-turn signals. On each side of D, there's one-turn signals. Upstream of J, you have a two-turn signal. This ensures that you always take a V, a D, and a J. You can never go V to J. You can't go V to C. You can't go D to C. You've always got to go V, D, J. So the cell always picks a variable region gene, a D gene segment, and a J gene segment at all times because it's got to obey this rule. Because that's the way the recombination system works. Now, this recombination, as I said, utilizes some of the normal enzymes that are involved in normal recombination in the cell. But it is a special recombination. There are some special genes that are required for this recombination. And two of those genes are the RAG genes, the RAG1 and the RAG2 genes. These code for proteins that actually, some of them recognize these sequences, the recombination signal sequences. So these are special genes that are required for just this recombination. Now, the reason I point that out is that there are, in fact, immunodeficiencies that are a consequence of deficiencies of either the RAG1 or the RAG2 gene. So if you had a deficiency of this RAG1 and RAG2 gene and couldn't carry out this recombination process, what would happen is you would never be able to generate functional B cells. You can't generate the receptor for antigen on B cells. So any cells that are there would be non-functional. Okay? So you don't have, these patients don't have B cells. In fact, these patients also don't have T cells. The reason they don't have T cells, again, we haven't talked a lot about the T cell receptor for antigen, but there is one, and it also has variable region genes and J gene segments and D gene segments. And the same story that I'm telling you about with the immunoglobulin, the heavy chains and light chains, also applies to the T cell receptor. And they use these same recombination signal sequences, and they obey the same rules of one-turn, two-turn signal recombination. And so if you lack any of these proteins, if you lack the RAG1 or the RAG2 genes, a deficiency that results in not only the loss of B cells, but the loss of T cells. And it's one of the forms of severe combined immunodeficiency disease, patients that have neither a T cell system nor B cells, no functional T cells, no functional B cells. And they result, there are several other forms of severe combined, other reasons for it, but this is one type of severe combined immunodeficiency, a deficiency in actually these enzymes or these proteins. Fine.
Is there any difference between your RAG1 or RAG2 or
Yeah, RAG1 actually is more critical. If you have RAG1, you basically have no B cells, no T cells. If you lack RAG1, if you lack RAG2, there are some residual cells. It's not as severe as lacking RAG1, but it is still a problem. I mean, you don't have very many good T or B cells. But yes, RAG1 is actually more important than RAG2.
You mentioned we go from B to J. Go from J.
In this case, in the heavy chains? J, well, in this one, of course, you can go J to V to J, right? Because it's a one and a two-turn signal. This ensures, what I'm just saying that you have to take a variable region and a J gene segment. Here you could never go V to J directly because there are two-turn signals on both sides, and you need a one and a two-turn signal to be involved in this recombination process. Right? So this would never occur. Here it can, here it can.
When you look at the germline gene organization, when you have all the variables, all the D regions, all that, do you still have, like, where do the recombination signals there?
All right, let's go see if I can go back and get one up to the germline. All right, yes, what would happen is all these recombination signal sequences, every V gene here is going to have a recombination signal sequence after it. Every D gene is going to a D gene segment is going to have them on both sides. Every J gene is going to have one here. Okay? All right. Any other questions? All right. Now, there is actually a strict order in which these genes are expressed as well. So when a B cell, for example, starts undergoing B cell development and starts committing to becoming a B cell, it expresses these genes in a particular order. And in fact, the first thing that it does is tries to rearrange the heavy chain genes. Okay? But now remember, we're diploid organisms. We inherited a set of these genes from our mother and another set from our father. So what happens is that the cell starts rearranging one of those, and it's random. Some cells may choose the maternal chromosome to start rearranging the heavy chains. Some cells, it's going to be the paternal genes that'll rearrange. But only one of them starts rearranging, okay? And so what we want to talk about is that this order of rearrangement and how the cell then ultimately expresses these cells. So what happens? So we have our progenitor B cell here. All right, and so we're diploid. So some cells are going to choose the maternal chromosome. Some cells are going to choose the paternal heavy chain genes to rearrange. So let's say that in our example, let's say that the maternal chromosome starts being rearranged. So we start rearranging the maternal chromosome, the heavy chain genes on the maternal chromosome. And it undergoes VDJ recombination. What the cell does next is to test whether or not that rearrangement was productive. And by productive, what I mean is that, you know, recombination, there can be mistakes made in recombination processes. So the cell tests to make sure that it did the recombination properly. And the way that it knows whether or not it was a productive or a good rearrangement is the cell is making at this time, remember there's no light chains now. We were doing only the heavy chain. So there's no light chain there. But there is a protein that is referred to as surrogate light chain. And if the cell is able to associate this heavy chain that it's made with the surrogate light chain, then it knows, yes, I can make, this was a good rearrangement, I can go ahead. So if the answer to this question was yes, that this is a good rearrangement, then what happens? That cell goes on and starts making mu and eventually delta, but there is a preference for making delta. So we end up with the cell that has now is expressing a maternal mu heavy chain. As a consequence of this productive rearrangement, what also happens is the cell turns off the rearrangement of the paternal chromosome. So the paternal genes never get rearranged. Okay? All right, so what happens if the answer is no? Suppose a mistake is made in that recombination and you make a heavy chain that can't associate with surrogate light chain. Then what happens? So if the answer to this question is no, then the cell goes on and starts rearranging the paternal heavy chain genes and goes through this process again. So it undergoes VDJ rearrangement. It asks, is this a good rearrangement? Which it knows if it can associate that protein with surrogate light chain. So if the answer is yes, we'll now have a cell that is expressing a paternal mu chain. All right? If the answer after the second time is no, that the rearrangement was in fact bad, that cell undergoes apoptosis and dies. It's a wasted cell. Okay? So what we end up with, either we have a cell that dies because it didn't rearrange either of the chromosomes correctly, or we end up with a cell that is expressing either a maternal heavy chain or a paternal heavy chain. And again, it starts with mu. So what started off as a progenitor cell now gets to a stage in B cell development that is referred to as the pre-B cell. This is a pre-B cell, is a cell that is expressing a heavy chain but no light chain yet. Okay? All right. If the cell makes it to this stage and doesn't die, if the cell makes it to this stage, then what it does. So now we have either this or we have either a maternal or a paternal mu chain being expressed in this cell. Then the cell starts rearranging the light chains. But we've got two light chains, kappa and lambda, and we've got two copies of each, one from our mothers, one from our fathers. And what happens is the first thing it starts rearranging is actually one of the kappa genes. And again, it's random. Sometimes it may be the paternal kappa, and sometimes it may be the maternal kappa. All right, so it starts rearranging the maternal kappa gene, say, and it undergoes VJ recombination, and then it tests, is this a productive rearrangement? The way it knows if it's a productive rearrangement now is, you've got heavy chain there. If it can assemble the light chain with the heavy chain and make an immunoglobulin molecule, assemble a complete immunoglobulin molecule, then the answer to the question is yes, this was a good rearrangement. And we end up then with the mature B cell that's going to express on its surface IgM and IgD. It's going to make only one light chain and one heavy chain. Okay? Two classes of that heavy chain, but one as far as specificity is concerned. All right, so if the answer was no, however, that the rearrangement was not good, then it goes around and starts rearranging the other kappa set of kappa genes. And it goes through this process again. The answer is yes, fine, you come down here. And also, whenever the answer is yes, you shut off rearrangement of all the other genes as well. If the answer is no, then you come around and then you start trying one of the lambda genes. You go through this process. Yes, okay, then you've got a lambda-bearing cell here. If the answer is no, you go around and you try the fourth chromosome, the second lambda chromosome, okay? And so basically what happens is a cell gets 2 chances to make a heavy chain and 4 chances to make a good light chain. If after the 4th chance making a light chain it is unsuccessful, it dies. It undergoes apoptosis. Okay? But what happens as a consequence of this, and we end up at this mature B cell stage, this ensures that a B cell only expresses one light chain and one heavy chain as far as specificity is concerned. It does have two classes, IgM and IgD, but they're the same specificity. They're going to be the same specificity because they have the same VDJ region. Okay? And now we have the mature B cell that expresses on its surface these 2 classes of immunoglobulin. Now, this order of gene, yes?
When you have like between combinations and things that were productive, you said that it would lead to this. Where is the light chain associated with the first part?
In the first,
The heavy chain.
in this one?
Yeah, after recombination,
In the heavy chains, it tries to associate with surrogate light chain. Surrogate light chain, okay? And then if that's true, then we know it's good. And then it goes on to do the light chains. Once the light chain is there, then it can assemble with whatever heavy chain that is making. Okay? Anything else? Okay? This order of gene expression, it really explains a lot of things. It explains why B cells only produce one kind of immunoglobulin with one kind of heavy chain, one kind of light chain. Again, as far as the specificity, because you're taking the same VDJ that was present on mu and D, associated with only one light chain that it can make, so the cell can only express one specificity at a time. It can never express more than one specificity. Okay? It also explains why there's allelic exclusion, because basically, because of the way the order of gene expressions, a B cell only expresses either a maternal chromosome or a paternal chromosome. So whatever allotypes are present are either going to be maternal allotypes or paternal allotypes. This also explains why the B cells, when they secrete immunoglobulins, secrete only one kind of immunoglobulin with 2 identical light chains and 2 identical heavy chains. Because any one cell only makes one light chain or one heavy chain. It doesn't make multiple chains. If you didn't have allelic exclusion going on, if you didn't have this orderly expression of excluding one allele and only expressing one, what would be the consequences of that? If a B cell could express both a paternal and a maternal, in other words, rearrange the paternal genes and rearrange the paternal gene, what would be the consequences of that? Think about it. Huh? No, I don't think that's good. But in fact, the failure rate is very, very high. There's a lot of apoptosis going on. This recombination process is not very good. Most of the cells actually die. What? Basically, what would happen is your B cells would express on their surface antibodies with different specificities. Right? But the paternal chromosome could give you one specificity. The maternal chromosome can give you a different specificity. It depends on which V, D, and J is being used, right? So you would end up with cells that express, you'd have receptors on the surface of your B cells against multiple antigens. Each B cell would have receptors against more than one antigen. And of course, the consequences of that would be, is if you're exposed to antigens, let's say that you have a B cell that can react now with antigen A and B. So you get exposed to A, it gets activated and it starts secreting antibodies against B. They're useless. You don't have any use for those antibodies. So it's very important that we have this allelic exclusion in the immune system because it ensures that there's only one specificity on every B cell. Okay? All right. Any questions on that? Okay. Now I want to talk about antibody diversity. And I'm going to give you, again, a little historical perspective on this thing, and then we'll talk about how we generate diversity. First of all, what is diversity? Our diversity, our repertoire, is nothing else but the sum total of all the antibody specificity that an organism is capable of making. The whole number of different kinds of antibodies that you can make, all right, is your diversity or your repertoire. This is different. Heterogeneity refers to a lot of different kinds of immunoglobulin molecules. Diversity refers to the specificities of the molecules. So the sum total of all the specificities you can make. Now, people make estimates of what our diversity is, and generally they fall somewhere usually in the range of 10 to the 7th to 10 to the 8th. So in other words, we have the capacity to make somewhere between 10 to the 7th to 10 to the 8th different antibody molecules as far as their specificity is concerned. In other words, we can make antibodies against 10 to the 7th to 10 to the 8th different antigens. All right? So the question arises, how is it that we can generate that much diversity? And so a lot of theories were proposed as to how we could generate the diversity. And there were a whole series of them, but basically they fall into two general categories. The one are the germline theories. And what the germline theories say is that we have in our germline the genes for all these specificities. In fact, the strict germline theory would say we would have to have somewhere between 10 to the 7th to 10 to the 8th genes that code for all these different antibody specificities. Now, at the time these were proposed, of course, people then tried doing calculations as to whether or not we had enough DNA, and most people felt that we probably have enough DNA to do that. Although it would take up a large part of our genome, would basically be devoted simply to making immunoglobulin molecules. Okay, so there was a lot of concern with these theories. The other extreme were the somatic mutation theories, and what the somatic mutation theory stated was that, well, no, we don't have a lot of genes. In fact, the strict somatic mutation theory says we only have one gene for the immunoglobulins. And what we do is we activate in the B cells a somatic mutation mechanism which introduces mutations in that gene. And it's the mutations that generate the diversity. Well, not surprisingly, the truth seems to be somewhere in between those extremes. Clearly, the germline theory is partially correct in the sense that we do have multiple genes for the immunoglobulins. We know we've got about 1,000 variable region genes in the heavy chains. We got about 300 or so in the kappa chains, and there's about 30 or so variable region genes in the lambda light chains. So clearly we've got more than one immunoglobulin gene. So they're partly true. But the somatic mutation theory is also true because there is, in fact, a somatic mutation mechanism activated in B cells. So what is our current understanding of where we generate or how we generate our diversity? Well, clearly, one of them comes from the fact that we've got multiple variable region genes. So we've got a number of different genes. That certainly contributes to our diversity. The main part of the diversity, however, appears to be coming from this VJ recombination in the case of the light chains and VDJ recombination in the case of the heavy chains. And the reason I say that is that it turns out that when V and J are coming together in the case of the light chain and VDJ is coming together in the case of the heavy chain, what you are essentially generating, that corresponds with these junctions where V, D, and J are coming together and V and J are coming together here, is the third hypervariable region. And if you remember back to that graph where we talked about the hypervariable region, you saw that was the most variable of all the regions. In fact, the third hypervariable region also contributes most to the combining site of the antibody. So what you're doing by making different combinations of V, D, and J, in the case of the heavy chain, or V and J in the case of the light chain, is you're generating different hypervariable regions, generating new antibodies of different specificities. So by mixing all these, making V1 next to D1, V1 next to D2, V2 next to D3, each of those generates an antibody of a different specificity because they have different hypervariable regions. So the bulk of the thing appears to be coming from this type of joining. Now, what also contributes to our diversity is something called junctional diversity. And this is just a fancy term. Well, basically, that's all it means is slight inaccuracies in the recombination process. And let me illustrate what I mean by that. Let's say that this is the V gene. The V gene is going to be in green, and the D gene segment is going to be in yellow, okay? And this is in this goldish color or tannish color. Those are the sequences in the intron. And I'm just showing you just 3 of the sequences. So this is that intron. So these are going to come together. V is going to come next to D, okay, in this recombination process. What's supposed to happen, of course, is the recombination is supposed to bring the V cut here and cut there and bring V right next to D, in which case you would generate a sequence CCC-TGG, which codes for proline tryptophan. So that, if that event occurred, you would have proline tryptophan in the third hypervariable region. All right? Now, look what happens if a mistake is made. And instead, if this recombination machinery just slips one nucleotide over, if it slips one nucleotide over and cuts here instead, as long as it slips one on the other side, you won't destroy the reading frame when the message is being translated into protein. So the reading frame will be maintained as long as you make the same mistake on the other side. So if this whole machinery slips over a little bit, you will cut here and cut there, in which case you generate a sequence CCC-CGG, which codes for proline arginine. If it slips over just 2 nucleotides, again, as long as it slips over on the other side, the same 2, you don't destroy the reading frame. Now you have a sequence CCC-CCG, which codes for pro-pro. So by slight inaccuracies and slight shifting of this recombination machinery, we can generate 3 different sequences, all of which are going to be located in the third hypervariable region. So these can specify antibodies with different specificities because the hypervariable region determines the specificities. So that's what junctional diversity is. The next thing that's thought to contribute to diversity is N-region insertions. So what are N-region insertions? If you sequence the protein, the immunoglobulin protein in the hypervariable regions, and you know what the sequence is. Now, if you go and then clone the gene that codes, the germline gene that codes for that immunoglobulin molecule, and then from the germline gene, of course, you can predict what the amino acid sequence is going to be from the sequence, right? Well, it turns out that oftentimes you find amino acids in the protein that were never encoded by anything in the germline. So here we have a germline that doesn't code for this, but yet the protein, the amino acids are there. Those are referred to as the N-region insertions. So these are amino acids found in the hypervariable regions that were never encoded in the germline. So where do they come from? Well, to look at that, we need to look a little bit more at how this recombination event occurs. So we'll look at a DJ recombination. So we're going to have a DJ recombination going on. What happens in this recombination is that first there is a cut on the D end so that you have free D, all right? But the intron is still attached to J here. So that break occurs first. Now, obviously, this is occurring in some machinery. This isn't going to be floating around free in the cell after that. It's all contained in there. But it is broken, and it is available. All right? Thing is going. All right. Now, in the cell at this time, there is an enzyme that is active, and it is called TDT. TDT stands for terminal deoxytidylnucleotide transferase. Okay? Terminal deoxytidylnucleotide transferase. Now, this enzyme has a unique activity. What it does is it adds nucleotides onto a primer, onto a free 3' hydroxyl group, using as a primer. But it doesn't have to have a template to read. In other words, it just sticks nucleotides in randomly, but it's got to attach it to some other piece of DNA. All right? Well, when this thing is broken, we have now got here a free 3' hydroxyl group. So TDT can come in here and add some nucleotides just randomly. All right? As the recombination process continues, we're going to cut here at J and religate these segments. And the intron is going to come out, actually, it comes out as a circle. So now we have, here's V, D, and J. And now we've got this sequence in here that was put in there by terminal transferase. This was never encoded in the germline, but it is there in the rearranged gene. That gives rise to those end region insertions. Again, so we see these end region insertions occurring as a consequence of these recombination processes, or during that recombination process. But this is going to be in hypervariable region 3, generating more differences, more variability in that third hypervariable region, adding to our diversity. Yeah, and if you did, that would be a non-productive rearrangement, right? And you probably couldn't even synthesize the whole chain. If you got a frame shift mutation, you probably wouldn't even synthesize the whole heavy chain then. And so it would be non-productive. And that's why I say there's a lot of errors made in these processes. In fact, most of the cells die off. Okay. Next, somatic mutation. It is clear now that B cells do, in fact, have a somatic mutation mechanism, and they can introduce mutations in the immunoglobulin genes. Because all the variability that we've been talking about so far, by VDJ recombination, junctional diversity, or end region insertions, explains how you get variability, the third hypervariable region. But it doesn't explain what's happening in the hypervariable region 1 and 2. And there is a somatic mutation mechanism that can introduce mutations. The mutations are not completely random. There are, in fact, hotspots where, in the gene where the recombination tends to occur. So somatic mutation adds some additional diversity. And then combinatorial association. This is nothing else but a fancy way of saying that a B cell can make any heavy chain and make any light chain, and when it assembles heavy and light chain, that's what ultimately determines the specificity. So even if you took this exact same heavy chain, but you combined it with two different light chains, you would get antibodies with different specificities, because the specificity is determined by both the heavy and the light chain. All right, so that's what is combinatorial association. And finally, multispecificity, again, just a fancy way of saying that any individual antibody molecule could potentially bind to more than one epitope. So it would have multispecificity. It would have it would not be completely specific. We have multiple specificities. All right? So these are the ways that our current concepts are understanding of how we generate the diversity. Now, to illustrate that, in fact, we can generate the diversity that we theoretically need, let's just go through this little exercise. Let's only consider heavy chains and kappa light chains. Forget the lambda light chains for now. Just to consider heavy chains and lambda light chains. There are 1,000 heavy chain variable region genes. There's 300 light variable region genes. Okay, you got 15 D segments, none here. 4 and 4. Okay, there may be 5, but let's say 4. Okay. All right, so the numbers of different possible combinations in the case of the heavy chain of V, Ds, and Js is actually the product of those numbers. So you can make 1,000 times 15 times 4, or 6 times 10 to the 4 different heavy chains simply by just mixing different combinations of V, Ds, and Js. Okay? In the case of the light chain, you can make the product of these, or 1,200 different light chains. By combinatorial association, since any one of these light chains could theoretically associate with any one of these heavy chains, the total number of different combinations is the product of those, or 7 times 10 to the 7. So we can get up to this, our anticipated 10 to the 7th to 10 to the 8th different specificities in our diversity simply by these mechanisms. We haven't even begun to consider the contributions of end-region insertions, junctional diversity, or somatic mutation in this process. So this VDJ recombination really gives the immune system basically a way to generate antibodies of a lot of different specificities so that we're protected against virtually anything that we can come in contact with. If we come in contact with an antigen for which we don't have a specificity, we just don't respond to that. We're non-responders to that particular antigen. Okay? Any questions? Yeah.
Does the model account for the products
That aren't useful?
that are not useful? Some of these are not. Yeah, that.
No, only if the rearrangement, a mistake is made during the rearrangement, will it be thrown away. If the rearrangement goes properly and you generate these, we have these things available. We will have all of those antibodies available to us. In other words, we will have a B cell in our body that expresses all of those possible combinations of heavy chains and light chains, just now sitting there. Okay, those cells that underwent apoptosis were cells that didn't rearrange the things properly. And so they couldn't make a good heavy chain or they couldn't make a good light chain. So they get wasted. But if you make a good heavy chain, if you make a good light chain, then you express that on the B cells. Anything else? All right. You may or may not have noticed that throughout this entire discussion, not once have I mentioned a role for antigen in this process. And that's because antigen plays no role in this process. This goes on completely independent of antigen. As our B cells are developing, this is going on. And we generate all this diversity. So somewhere in our lymph nodes or spleen or somewhere in the body, we have a B cell that is expressing all the has on its surface an IgM and an IgD molecule that has specificity against everything that we can possibly ever make antibodies against. They're not secreting those antibodies. They're B cells with that immunoglobulin on their surface just waiting. Okay, so all of this is independent. So what role does antigen play? Antigen selects out those clones of cells that have the appropriate receptor, so that it can bind to. That cell will then undergo its terminal stages of differentiation into a plasma cell and ultimately secrete antibodies. In the case of T-independent antigens, they do that without any T cell help. In the case of T-dependent antigen, they're going to require some T cell help in order to differentiate into those plasma cells. But eventually, so what antigen does is antigen selects out the appropriate clones of cells that have the appropriate receptors for that antigen. That then activates the cell and undergoes final stages of differentiation and becomes an antibody-secreting cell. The antibody that is secreted by the cell when it is a plasma cell has the same specificity as the immunoglobulin that was sitting on that original B cell that bound the antigen and got activated. Okay, so the specificity will remain the same. Okay. All right, let me just finish one last thing and then we'll take our break. Again, we haven't said a lot about the T cell receptor. Dr. McCallop will tell you a lot more detail about it, but there is a T cell receptor for antigen. It's composed of two chains. They just happen to be called alpha beta instead of heavy and light because they're roughly the same size. I just want to point out that the gene structure of the alpha chain is identical to the immunoglobulin light chain. In other words, it has V's and J's, and it does everything that we just described for the light chains. Okay. The gene structure for the beta chains is like the heavy chain. In other words, it has V, D's, and J's. And it does the same thing that the heavy chains do, okay? So everything I just told you about the heavy chain, the immunoglobulins, also applies to the T cell receptor genes. With one exception, there's no somatic mutation in T cells. Okay? Okay, we can take our break now.